Alright, everybody. Welcome to Lecture 21. Almost done with the quarter. Um, Thankfully, I am getting pretty burnt out. But, here we are. We're going to finish these up and, yeah, go from there. I think there's, after this one, there's 3 or 4 more lectures that need to be done, and then we're done until the final. So, yeah, let's get into it. So in the previous lecture videos, we kind of talked about synthesis of fatty acids and distribution a little bit. So today's going to be a little bit different on this, along the same lines though. So triglycerols are a long-term energy storage, right? So basically, you store them in your blood, or not blood, but you store them in your fat cells in your liver, and you can use them later. And in terms of energy, they actually provide a lot of energy. So triglycerols are like 37 kilojoules per gram, and carbs, so sugars, are like 17. It's pretty much over double for a triglycerol. Um, so yeah. And in terms of storage, triglycerols are stored in like these fatty, insoluble blobs, right? And glycogen, on the other hand, has to be dissolved in water. So it's 2 grams of water per 1 gram of hydrogen. So all that to say that triglycerols store a lot more fuel than like glycogens and sugars like that. So in terms of like when you shift, basically, and we've already talked about this, like in order to get the energy out of the triglycerols, you have to hydrolyze them into acetyl-CoA groups to put them into the citric acid cycle. So you have to hydrolyze them, and then you have to oxidize them into acetyl-CoA, and then, yeah. That basically, what that allows for you to do is it allows you to preserve glucose for the brain. It also allows you to preserve proteins, so you don't start breaking down proteins. And it also allows you to preserve amino acids, so you also, nucleic acids, so you don't break those down to hopefully produce sugars out of them. Because what happens is if you run out of like free glucose or whatever, your body will try to break down whatever is available. So it'll try to break down proteins and stuff like that. But if fatty acids are present, it'll actually break down those first. So funny enough, actually, 40%, more than 40% of the daily energy requirements for humans is supplied by triacylglycerols. And it's just a lot of it. It's like 50 to 80% of daily energy requirements for liver. And it's basically the sole source of energy for hibernating animals, which should make sense, right? Because hibernating animals are not getting glucose from the environment, so their stored fat is what allows them to hibernate. So, in terms of mobilization, we can talk about this a little bit. So, fatty acids, right, in order to be moved around the body, they need to be mobilized from adipose tissue, so fat cells, and transported to other tissue to use as energy, right? So, it has to be transported in the blood. So the question is, how are they degraded and how are they oxidized to yield ATP? So I mean, the couple steps are, first we have to mobilize the fatty acids so that they can leave the adipose cells. Then we have to activate these fatty acids, turn them into acyl-CoAs, because remember, these are actually in the cytosol, right? But you'll see later that breakdown occurs in the mitochondria. So we have to do that. We have to transport them into the mitochondria via carnitine shuttle, and the carnitine shuttle is the thing that leads to this acyl-CoA, basically. And then we have to do beta oxidation, which is the breakdown of them. And these should hopefully make sense by now. Glucagon, right, stimulates you putting glucose into your blood or producing energy. So that would suggest that it would promote breakdown of fatty acids, right? So you can use them. So glucagon will bind, right, or the glucagon hormone will be released, right? It'll bind to a GPCR, and glucagon, remember, leads to phosphorylation. So it'll lead to the activation of this cyclase, which stimulates cyclic AMP, which activates this pKa, and then that pKa, right, phosphorylates your beta oxidation enzymes and stuff like that. And basically it promotes breakdown. It also phosphorylates these cytosolic hormone-sensitive lipases, which help break down the and these lipase, remember, is something that breaks down fats or triglycerols, or triacylglycerols. So this is very important because this basically stimulates this whole process to break down the fat cell, or not the fat cell, to break down these fat globules in the fat cells, so they can release the you can release the fat into the blood so that other cells can pick it up. So, you know, PKA phosphorylates paralipins, and these paralipins kind of help with, you know, keeping the fat globules like collected together and blocked up. But when you phosphorylate them, they basically dissociate, and it allows for the fat to be broken down pretty easily, and stuff like that. So hormone sensitive, or these hormones trigger the mobilization. We talked about this before. So dissociation of paralipins comes from the phosphorylation, and it results in the activation of a couple things. And I can't actually remember the exact definition of the ATC, the ATGL. I'm not gonna lie. But basically, this is I'm pretty sure it's a type of lipase, and so it breaks down. That one is responsible for breaking down triacylglycerols, so it breaks it into a diacylglycerol and a free fatty acid, and then the HSL, the hormone-sensitive lipase, breaks down diacylglycerols, basically. I can't remember what it is, actually. I don't know what the A stands for. I think it's supposed to be triacylglycerol lipase. I think is what the TGL stands for. So TGL, HSL, and then MGL is monoacylglycerol lipase, I'm pretty sure. But you get the idea, basically. So basically, the dissociation of these paralipins after phosphorylating them leads to the activation of this, and that's activated through this like binding protein. So this basically, after the paralipin dissociates, it basically is free to move over here to activate the ATGL. And then from there, it goes from triacylglycerol to diacylglycerol. And then the hormone-sensitive lipase that's activated by PKA, right here, catalyzes diacylglycerol to a monoacylglycerol and free fatty acid. And then the MGL, the monoacylglycerol, catalyzes the release of the glycerol into free fatty acids. So you release all free fatty acids from this thing. And then there's also a couple other things. So fatty acid binding protein. This thing helps with transport fatty acids, free fatty acids within cells. So you have to bind these here basically. So otherwise they're just floating around. So there's a fatty acid binding protein that helps with the dissociation and transport of these things. And once they get into the blood, this is very important. Also very important for the MCAT if you intend to study for that. But eventually these free fatty acids bind to serum albumin in the blood, which is a protein, and that allows it to circulate around the blood and get transported into cells that need it. And so they enter the cells via transport proteins, and then they undergo beta oxidation to yield acetyl-CoA. And then eventually, the glycerol circulates back to the liver, and then it re-enters glycolysis, gluconeogenesis, whatever. Because glycerol, remember, is like a isomer of a glyceraldehyde 3-phosphate, so it can go back into gluconeogenesis or glycolysis or whatever. So basically, right, now that once we get these things into the cytosol, we have to get them into the mitochondria. And so, give me 1 second. As is whenever I do these videos, my mouth gets extremely dry. But anyways, as we need to get these into the mitochondria. So these short and medium chain fatty acids, so less than 12 carbons, these things can enter the mitochondria via diffusion. This is very important to keep in mind. So if it's below 12 carbons, it's considered a medium to small chain fatty acid. And because of how small they are, they're able to just enter the mitochondria freely through diffusion. And then they can just undergo beta oxidation and you're done. But for longer chain fatty acids, such as palmitate, that's like a 16 carbon, or anything greater than 14 carbons, these need to be activated and turned into acyl-CoAs. Because remember, technically these free fatty acids, they have this O- group, right? Or this OH right here. That OH is deprotonated at physiological pH, right? It becomes a minus charge. So when the chain is pretty small, the size and the fact that it's mostly hydrophobic make it so it is able to transport through, but at the larger sizes, the size plus the charge make it pretty difficult. So even though it's still very hydrophobic, it's still extremely large, and it's very difficult to transport that through the membrane through diffusion. So basically what we do is we activate this free fatty acid by turning it into an acyl-CoA molecule. And we do that through acyl-CoA synthetase. And it's formed on the cytosolic side. So, we're on the cytosolic side here. We turn it into acyl-CoA synthetase, and then we send it into the mitochondria. So here's kind of like the scheme for how we do that. We have a free fatty acid. We attach coenzyme A to it. And because it's a synthetase, it uses ATP to do so. So hydrolyzes the first phosphate bond, right? Then it hydrolyzes to a pyrophosphate. And then that pyrophosphate is cleaved again for an extra 30 kilojoules or whatever. So eventually we get this thing. And this is like an activated high energy fatty acid, right? So from here, once they're activated, we have two options. We can either synthesize and use them to make triacylglycerols, or we can break them down via beta oxidation. So in this case, right, you can imagine, right, maybe you want to synthesize a fat, right? Because remember when you used a fatty acid synthase, there was like an intermediate that looks like this, right? So, it kind of depends on what you want to do. So in this case, we're talking about beta oxidation, so we're going to talk about the carnitine shuttle. So the carnitine shuttle transports long fatty acid chains from the cytosol into the mitochondrial matrix, and it is the rate limiting step in beta oxidation. So all the other enzymes are like pretty quick, but this is the one that takes a bit. It's the transport time that takes a bit. So basically, to get this thing to work, we need to do transesterification of fatty acids to carnitine via carnitine acyl transferase, so CAT1. So this is different from this right here, right? This is acyl-CoA synthetase. This is to activate the free fatty acids. So now what we do is we take our fatty acyl-CoA that we just made, we add carnitine to it, and basically we're displacing this CoA. It's a transesterification reaction. And so once we do that, we make this fatty acyl carnitine, and once we make this fatty acyl carnitine, this can now diffuse through the outer mitochondrial membrane, but it does that through this transporter here. And so the way that it basically does it, this is a great scheme actually to study. Basically what's going on is you see our long chain fatty acid. It's going to be activated by acyl-CoA synthetase. And then once you activate it, you can either turn it into a lipid or whatever. You can turn it into a fatty acid or whatever and keep in the cytosol. Or we talk about breakdown. You can add carnitine to it using CAT1, carnitine acetyltransferase. That kicks off this CoA group, and you make fatty acyl carnitine. And then there's a translocase protein, and what this protein does is it's like an antiporter. As the fatty acyl carnitine goes into the mitochondrial matrix, a carnitine has to go out in the same way. So carnitine has to go out while the fatty acyl carnitine goes in. And then once we're in the mitochondria, we use a carnitine acetyltransferase with another CoA from the mitochondria, to reform this acyl-CoA and kick off this carnitine. And then that's what ends up producing the carnitine to go back out here in the first place. Because remember, this is like a process that's it's not happening with just one fatty acid at a time. It's happening with like hundreds of thousands of them at the same time, basically. This is what I just talked about, basically. So translocation across the inner mitochondrial membrane, the carnitine shuttle transferase. I guess I should be very specific here. The outer mitochondrial membrane, so this one technically is it's permeable enough so that these acyl carnitines can actually diffuse through it. It's just the inner membrane where it can't really diffuse through. So this image is a little bit misleading. This channel really should be like here. It should be on this membrane because it technically can diffuse through the outer membrane. So translocation of fatty acyl carnitine across the inner mitochondrial membranes. We talked about this via carnitine transporter, the translocase. And we already talked about how the transesterification of fatty acyl to CoA, right? Basically regenerates the fatty acyl CoA, and it regenerates a carnitine, right? And so carnitine shuttles back into the cytosol via this translocase as well. And so then we can repeat this process over and over again, right? Because now CAT1 can use Cat1 can use this carnitine that we just brought out and stuff like that. Now we can talk about beta oxidation. We'll talk about this in terms of the 16 carbon palmitate, I guess, based on this lecture, but everything else is pretty much the same. So the main thing to keep in mind is, for a 16 carbon palmitate, there are 7 cycles of beta oxidation, and in total it will yield 8 acetyl-CoAs, 7 FADH2s, and 7 NADHs. And basically, I hope you're able to see it's kind of like the reverse of the synthesis. 8 acetyl-CoA should hopefully make sense. There's 16 carbons. Each acetyl-CoA has 2 carbons. Therefore, you're going to get 8 acetyl-CoAs because 8 times 2 is 16. And basically, each of these acetyl-CoAs, in order to be made, requires 1 FADH2 and 1 NADH. And, basically, when you're basically making NADH and FADH2 here, and you're also making it when you go through TCA and eventually use it in an electron transport chain, right? So each acetyl-CoA makes, I think it's 3 NADH and 1 FADH2 on top of that. So you can imagine that you're making a lot of electron carriers here. And so, like I said, electron transport and stuff like that. So here we can talk about kind of what's happening here exactly. So step 1, in beta oxidation, there are 4 different enzyme catalyzed reactions. And there's a slide later I'll talk about in a second. But for even-numbered, full-fat saturated fatty acids, this process is pretty straightforward. We start with a dehydrogenase. So acyl-CoA dehydrogenase. I wouldn't say it's super important to remember the exact enzyme names. The general steps is definitely the thing you should keep in mind, but if you want to write the names on your note card, I wouldn't be against that. So the first thing we have to do is we have to do an oxidation reaction, and so this oxidation reaction uses FAD as its substrate and the fatty acid that you're using. So you can see here, right? Look at this alpha and beta carbon. What basically happens is you use FAD and you generate FADH2. And this FADH2, right, can then go into, electron transport chain, all that stuff. And so when you make this FADH2, you can see here now, you look at these 2 carbons, right? You see there's a double bond that's formed here. So you've removed 2 of the hydrogens, and you form a double bond. Perfect, that should make sense. Then you do a hydration reaction where you add H2O to this double bond. So you add this H2O, and you can see the H2O is added to this side. So it's added to not to this carbon, but it's added to the carbon next to it. So basically it's 1 carbon away from this carbonyl. So 1 carbon away from this carbonyl. From there, we then oxidize it. So we oxidize it using NAD+ now. So NAD+ is used to oxidize this alcohol to a carbonyl. And then we do I think it's not a Clemmensen, what's it called? I think it's a reverse aldol, basically, right? Where now we do another transesterification, where we use this CoA. This CoA attacks here and basically kicks off this whole group. And so you can see here, we now form an acetyl-CoA, right? From the base, right? Now, because the base was where we started, so this is supposed to be the base right here, right? So the base gets kicked off as acetyl-CoA, and this new base here is the remainder of the fatty acid chain. So like I said before, we oxidize to generate NADH, and then we use acetyl-CoA acetyltransferase, so thiolysis, right? It's a transesterification. It's a cleavage reaction. And it results in a fatty acyl-CoA that's shortened by 2 carbons, so the fatty acid chain, and 2 carbons released as acetyl-CoA. And then you would do this process all over again 7 times until you get rid of the entirety of the chain. So you'd produce 1 acetyl-CoA, 2, 3, 4, 5, 6, 7, and then 8 acetyl-CoAs, right? And so here is a nice little image talking about where these things occur. The ones I would keep in mind for sure are the location. So fatty acid synthesis is in the cytosol. Oxidations in the matrix. In terms of synthesis, the enzyme that does it is fatty acid synthase for synthesis. It's one enzyme, but it's separate individual enzymes for oxidation. And stuff like that. In terms of fatty acid synthesis, remember we actually use NADPH but then in oxidation we use NAD plus and FAD. And then, for carbon units, we add 2 carbons in the form of malonyl-CoA, right? So we activate it, and then you yield 2 carbons in the form of acetyl-CoA. So you can kind of see this image. This is a nice table, I think, that you can use to kind of study and understand what's going on. So I would say for sure keep this table in mind while you're studying. So this is just to show you that it's basically just trying to show you that these mechanisms for this enzyme, these enzymes for the beta oxidation enzymes are conserved mechanisms. So you can see like this beta oxidation one, how it uses FADH2. It's the same thing for succinate dehydrogenase, right? Succinate dehydrogenase does the exact same thing. So it's like a beta oxidation. What it does is you use FAD to remove 2 H's, and then you put a water onto it, right? And then you oxidize it. In citric acid cycle, we go from succinate to fumarate to malate, right? So we basically do that. And then similarly for an amino acid, like such as isoleucine and leucine and valine, you do the exact same thing. You remove a couple H's, you hydrolyze it, and then you oxidize it, right? And then you do a couple things like that, and you get a product that you can use in citric acid cycles. So here's a slide that shows you the energy yield. So 16 carbon palmitate would go through 7 cycles of beta oxidation, producing 7 FADH2s and 7 NADHs. And remember from 440 if you can, 1.5 ATP per FADH2 and 2.5 ATP per NADH. So from these 7 FADH2s, we get 10.5 ATPs. From the 7 NADHs, we make 17.5 ATP. And then remember, each acetyl-CoA makes 3 ATP and 1 FADH2, right? So you basically do this whole thing where you're doing a bunch. So you're going to make 10 ATPs per acetyl-CoA. And there's 8 acetyl-CoAs, so you use 10 times 8, and you get 80 acetyl-CoAs from this. So you make 106 ATPs for every palmitate that is oxidized, basically. So that is a lot of energy. So fatty acyl-CoAs within the cytosol, they have 2 main phases. We've talked about this. They either remain in the cytosol for the creation of phospholipids and stuff, or they are oxidized to use as energy. Basically, the important thing about like the fatty acyl carnitine shuttle is that if you attach carnitine to the fatty acid, it commits it to going through oxidation. And the other thing that's important to know is in inhibition. Malonyl-CoA, the thing that you use to synthesize fatty acids, is actually an inhibitor of Cat1. So Cat1, remember, is the thing that adds the carnitine onto the acyl-CoA, right, when you activate it. So that's where like the regulation occurs. And this basically what it does is it, like it says here, prevents breakdown and synthesis at the same time, because otherwise you're just wasting energy. So here is like a little bit of I think just a more regulation. So when glucose levels drop between meals, glucagon is released. Because you have less sugar and less energy. So what this does is it phosphorylates things, right? So it inactivates acetyl-CoA carboxylase, which inhibits fatty acid synthase. And because you have dropped the activity of acetyl-CoA carboxylase, that leads to less malonyl-CoA, right? Which, because malonyl-CoA inhibits Cat1, when you get rid of it, Cat1 becomes active, and so that triggers the mobilization of fatty acids in adipose tissue and stuff like that. So this is an image of that diagram if you want to look at that in more depth. But hopefully that makes sense. Essentially, it's like the hormone itself doesn't directly affect Cat1, but it affects malonyl-CoA levels, and malonyl-CoA levels themselves do affect Cat1 activity. So ketone bodies. Ketone bodies are produced in the liver, and they do this when glucose is scarce. Specifically, glucose is scarce. And the body needs to, replenish these levels of triacylglycerols. This happens, when you're on when you're fasting, where you're on a ketogenic, so a low carb diet, so not really much sugar. Also, it occurs during prolonged exercise, and there's like diabetes as well that occurs. This is like a really important process in the brain, because glucose is the primary energy source for the brain. The brain uses like 60% of your glucose. It's insane. So ketone bodies, what they do is they're formed from acetyl-CoA. They're kind of like fatty acids, but they're not really fatty acids. And they happen when oxaloacetate is shunted towards gluconeogenesis. So this is kind of like an example of that. So like you got a couple here. So acetoacetate, beta hydroxybutyrate, acetone. These are basically transported by the blood to other tissues. And then remember, this is when carbohydrates are low. So this is when oxaloacetate is being pushed back into gluconeogenesis, right? And so you basically you add all sorts of extra things onto these carbon chains, and then you shuttle them out. And so these are transported by the blood to other tissues, and for the conversion of and they get converted to acetyl-CoA in other tissues, and then they get oxidized for ATP. But then acetone actually is a byproduct that's exhaled. Actually, so acetone is exhaled. I think if I'm not mistaken, these are the two that you actually see more often in terms of ketone bodies. This is just exhaled out. For untreated type 2 diabetes, this diabetic ketoacidosis is a really interesting disease. Basically what happens is, your insulin levels are high, but there's no intake of glucose. And so when there's no intake of glucose, it's kind of like your cells are starving. And so, you don't really... You're trying, because you're trying to... If there's no intake of glucose, right, you can't make acetyl-CoA, and so you can't make malonyl-CoA. And so... because there's no malonyl-CoA, that relieves the inhibition on CAT1, on carnitine, this thing. And so the cell thinks that there's not enough glucose in the body, and so it tries to oxidize fatty acids in the mitochondria. And so that ultimately results in a lot less TCA intermediates, because, all these acetyl-CoAs are being used to... form fatty acids. So basically you're storing energy that you don't need to store, and it just results in ketone bodies, because ketone bodies are far basically they're fatty acids but way smaller, and so you make these ketone bodies, and these ketone bodies, right, they have acidic groups on them, right? You can see like here these acidic groups, right? So these acidic groups here, they donate protons, and so it actually leads to a drop in blood pH. Because you're forming so many of these ketone bodies, and they're protonating everything, and then you get an acidic blood, basically. I honestly think that's really it. This is a bunch of this is like just a review. You can feel free to take a look at these if you'd like, but I don't think these are the most important slides. They're nice summary slides, but I feel like I've already covered them pretty in depth. So I'll let you guys take a look at these yourself. I'm trying to see if there's anything I would really care about here. Not really. Yeah, this is pretty much all what we just covered. So yeah, I would say that's it for this video then. And yeah, happy studying.